Okay, so thanks for joining us today. So I'm Joe from American Cancer Society. We've got two ACS grantees who had some really high impact papers just came out. Congrats to both of you. And thought it'd be fun to get the two of you on so you can kind of explain your work to each other, ask a few questions, and get to it. So, um, quick introductions. Dr. Gardini, do you want to?
Absolutely. Thank you very much. So my name is Alessandro Gardini, and I'm an assistant professor here at the Waystar Institute in Philadelphia. And I started my lab about, well, 6 years ago. That's right, about 6 years ago. And I actually became an ACS grantee starting in 2019. And yeah, generally speaking, my lab work revolves around transcriptional regulation and how the epigenetic landscape affect transcription in physiological conditions and in tumor cells.
Sounds good. And Dr. Huh, could you introduce yourself?
Yeah, of course. Thank you for this kind invitation. So, my name is Yuan, and I think I'm still assistant professor, but I got the good news that I'm promoted. So, but not until September. So, I guess it's in a transition from assistant to associate professor at Northwestern University, Department of Molecular Biosciences. So, my lab is interesting on the understanding molecular mechanisms by which large multi-subunit complexes engage in DNA-centric processes using a combination of biochemistry and structural biology tools. The main tool we're using is single particle cryo-electron microscopy, which gained some attention in recent years, offering high-resolution visualization of these amazing large complexes. I have another kind of topic. My lab is interested in DNA damage and sensing, but for this conversation, we'll probably just focus on gene regulation.
Right on. And sorry, I got your last name wrong. It's Dr. He, not Dr. Huh. I apologize.
But well, it's actually He, but it's just this confusion. I'm used to, telling people he,
Okay, I got you. Well, so you just had a paper in Science, and Dr. Gardini, I guess we'll start with you. You just published work in Cell recently. I'm going to read the title, and I'll turn it over to you. So it's the PP2A integrator CDK9 axis fine-tunes transcription and can be targeted therapeutically in cancer. Did I screw that up? Is that right?
Yeah, no, absolutely. You said it right.
So it's a bit over my head. That's okay. I'm not the audience. I think our friend Dr. Huh here is. So take it away. Tell us about your work.
Absolutely, and let me help myself here with like one slide. I hope you guys can see it. Do you? Okay. So really, in just a couple of minutes, I wanna sort of say what is the take-home message and why we're really happy about this discovery. And first of all, let me tell you that this has been an amazing collaboration, an amazing collaborative effort with the Johnston Lab over at Melbourne. We really have sort of lifted each other work in an amazing way. And but the main thing for us, so my lab is interested in understanding better the function of this large, speaking of large complexes implicated in transcription, like Yuan was telling about will tell us about mediator. Integrator is another one of such complexes. It's a 14 subunit complex that we know right now that it has multiple roles in transcriptional regulation. And what we basically published with this paper is that we identified yet another role that was previously unknown for the complex. So we have sort of isolated a couple of subunits of this complex that recruit a phosphatase. Now, phosphatases remove phosphate groups, and they're very interesting sort of complexes to study because they are apparently very simple. It's a small complex. The basic unit is just like 2 subunits. So people have been wondering for a long time, like what is the specificity in all this? How is specificity brought about in PP2A? And now what we understood with our paper is that these subunits of the integrator complex, they bring in the PP2A phosphatase at active sites of transcription in human cells, but this process obviously is conserved throughout lower organisms as well. And what we were able to show is that this comes in stark opposition with like sort of one of the dominant kinases in transcriptional activation in general is CDK9, which here is depicted, you know, the scientific name truly is like PTFB, as you can see here in the cartoon. But CDK9 is critically implicated in a process that's called, you know, pause release and elongation. And what I mean by pause release is that generally RNA pull 2, when transcribed genes, finds itself stuck at some point in a so-called pause conformation and needs the CDK9 activity to be released into an active elongation. Active elongation form means that we can actually transcribe the gene in full, have a messenger RNA that can be then, you know, exported to the cytoplasm and translated. So this is really a key point for us to proper expression of virtually any gene in a cell. So we were able to show that CDK9, it's not just about recruiting CDK9, it's about balancing. It's a balancing act in a way. Elongation is a balancing act between the CDK9 activity and the phosphatase activity of PP2A, which is brought over by the integrator complex. And we were able to characterize that at chromatin, this PP2A targets sites such as the C-terminal domain of RNA pull2 at its serine 2 at the serine 2 mark, and other sites such as the DSIF and so on and so forth. The bottom line, it's really the activity. I was stressing out the, you know, the balance in between the two, because we also found in the paper that, you know, a lot of tumors are, so to speak, transcription addicted. They kind of rely on constant pause, release, and elongation to boost their growth. So what we have found is that there is a great window of therapeutic opportunity in here. People have previously thought about using CDK9 inhibition. So using CDK9 inhibitors to curb transcription and curb tumor growth. When we now combine with some success, I mean, definitely there is some success in preclinical models, but what we found is that when you combine CDK9 inhibition with activation by small molecules of the PP2A phosphatase, what we're able to do is we increase this effect here. So we increase the recruitment of PP2A at chromatin, and we get a much better block on transcription in these tumors. And I guess I'll stop here.
So Dr. Gardini, that was great. Dr. Ho, when you, as you listened to that, as you read it over, did anything jump out at you, or do you have any questions you'd like to run past them?
Yeah, it's, you know, one question is, by the way, this is fantastic work. Congrats again. And it's this kind of a mystery of dual function of integrator. Because it's very surprising to see you have a phosphatase incorporated into this mysterious integrator complex that used to be thinking as a RNA processing enzyme, right? So what's your thought on, you know, integrating that kind of information into your model?
Absolutely. No, it's a great question. And to be fully honest, there's still so much that we don't understand. And I was the first one surprised, because I always, you know, I have worked on this complex for several years. Like it's been almost 10 years at this point. And I always thought about the whole overall, the complex as a transcriptional co-activator, right? Mm-hmm. Which is what we originally also proposed back many years ago, that the catalytic activity, like the RNA endonucleolytic activity, would do. So now, surprisingly, and structurally, you know, you've probably seen that a structure of integrator has come out recently, and indeed, we're talking about two very distinct modules here. They're not even close to one another. A phosphatase module is on the one hand, and on the other end, we have the RNA catalytic activity. So it's still, and I still think, you know, the modularity could be that, you know, we don't really have a fully assembled integrator complex. at any given time. So this could help explain a little bit while we have two activities that might be going against each other paradoxically, or they might actually be coordinated. So this is something that we are investigating right now.
Great. And if you don't mind, I actually have another question.
Absolutely.
What do you think about this kind of a tug of war of phosphatase competing with CDKs for the CTD of the polymerase throughout this kind of narrow window during the initiation phase of transcription? We're talking about potential competition with CDK7, but most likely in this picture, you're thinking it's the PTAB CDK9. So what do you think about like the role of integrator, this new information of having a phosphatase in this narrow window of event, where is most likely kind of in your picture of model, like how does it cooperate with the polymerase and potentially initiation machinery and transition into the pausing phase?
I'm thinking, I don't know, it's this idea of the balancing, this idea that just like, any enzymatic activity just left by itself could be counterproductive at some point. I think this is really the key probably to interpret the whole thing. So CDK9 by itself, so polymerase needs time to recruit additional factors, needs time to make sure that everything is there for a proper productive elongation, right? And I would say that the phosphatase brings that chance, bring the chance of like a sort of an additional layer. We're kind of stopping things. We have this active opposition every single time and I would say that this is probably happening at every single stage of transcription, right? Because at initiation and pause release. But one of the data that was very exciting to me to see is that we can chip, so we can actually find this phosphatase being actively engaged with RNA-APOL2 throughout the process of elongation and termination and post-termination.
Interesting.
Right? So there really is a constant need of not letting one activity take over too much and too soon, I guess.
Okay. So essentially, you think the integrator compound is providing almost like a platform to specifically locate the phosphatase into into the elongation complex.
I think it would also provide additional regulatory activity and not just like the sheer platform. But again, we need a few more years to get into that.
That's very interesting.
Right, this is fun. But why don't we switch gears? So from cell to science, from one high-profile discovery to another, Dr. He, you just had a publication in Science, structure of the human mediator-bound transcription preinitiation complex. There's a lot of syllables. So tell us all about it. Let's hear it.
Sure. Is there a way to share screen?
Yeah, if you look towards the top of your screen, I think you'll see a few icons, a camera, a mic, and a little rectangle with an arrow.
Yes.
If you click that,
got it.
Then you could either share your desktop, which can be dicey, or there you go. Looks good.
Can you see it?
Yes. I see the full. I see the slide, and I see the full.
What about now? Do you see the single slide or the presenter view?
I don't see the single slide. I can see it. It's a little smaller because I can see off to the left, the 4 slides. I can see the.
Wait.
It's not full screen.
Really? Okay. Sorry about this.
Oh, it's okay. I mean, I think I can see it.
What about now? Do you see the movie playing?
I do not.
No.
That's the problem. What about now? Can you see this? What happens here?
Teams. I wish we had Zoom.
still not in full screen. So I see you're hitting play, but you're still not in full screen, so kind of nothing really moves.
Interesting. Okay, so just, to save time.
When you shared, did you share your desktop or did you share the program? And maybe try doing the other one.
That's right. Okay, so I exited and then let me share the screen. Now you see my full screen, right?
Yeah, now I see.
See your
oh, there we go.
Okay.
Now, do you see the movie playing?
We see
like 2 slides.
View.
I see 2 slides, current and next.
What about now?
I see 1 slide.
Now it's perfect.
Okay, so I don't think it's playing, but anyway, now you see the slide. So
yeah. Sorry about that.
No worries. I think there's some kind of a limitation. I think I somehow had to like reboot and then restart and then give the permission for it to record or something. Anyway, so yes, we were able to use single particle cryo-EM with the help of 2 very talented graduate students in the lab, able to reveal for the very first time how human mediator, a 26 subunit complex, engage with the preinitiation complex shown here. So the colorful one on the top is the mediator. POL2 is colored in light gray, whereas all the GTFs, the general transcription factors, most of them are colored in this kind of a dark green color, and then TF2H core is light pink. From this structure, I'm not going to dive into the architecture of mediator, given the topic of this this conversation, mostly focusing on CDK7 cycling and MAT1, this so-called CAC module within TF2H. So 2H has 10 subunits. 3 of them are formed in this very flexible CAC module, and we were able to show that at very high resolution that how mediator complex through multiple modules engage and stabilize the CAC module of TF2H from the other angle, and then position the active site of the CDK7 right along the path of the CTD of POL2 to be able to phosphorylate CTD at serine 5, a critical modification to allow POL2 to escape the promoter and transition into an elongation mode. So, kind of echoing Alessandro's point on, these large complexes and kind of working together with other modifying enzymes, in this case, a kinase that is critical to establish the platform for the polymerase to allow elongation. So we were able to see, it can play actually. So it actually plays. So you can actually see the DNA engage with the TF2H. One of the key point is to be able to allow DNA translocase to thread the DNA into the active site of the POL2 to allow bubble opening. And we just mentioned about the CDK7 cyclin to be stabilized directly by the mediator complex to facilitate the positioning of this the CTD of the complex. I'm going to just very quickly go through this. This is not kind of related, but interesting point. What we notice is that a lot of these activator molecules that binds to enhancers likely not to have a stable interaction or like a rigid interaction with the main body of the mediator, but rather through these multivalent interaction network to bring copies of, multiple copies potentially of mediator to near the promoter region to allow the loading of the polymerase. This is zooming into the active site of CDK7 cycling H, and we can clearly see a peptide of RPP1 which, by the way, purified endogenously from HeLa cells through a collaboration with Bob Tijan's lab at UC Berkeley. And we were able to build a model based on consens sequence with other previously published crystal structures of CDK, like in this case CDK2, and they seems to share a very similar substrate recognition pattern. And this eventually allowed us to build a model of how this gigantic, 57 subunit machinery recognized CTD from the polymerase, and being able to phosphorylate in two distinct mechanism. We don't know, we cannot tell which mechanism is preferred. One is like the CDK7 is going to search and then find a unphosphorylated CTD and then continuously phosphorylate toward the C terminus, whereas the other one is the opposite reaction. So that requires further experiment to tell. And I'm not sure structural biology is the best way to actually figure this out. And with that, I think that's pretty much it.
Right on. Alessandro, what do you think?
It's an amazing work, and I love this modeling of how the CTD phosphorylation is happening.
1 second, you may want to stop sharing your screen.
Sure.
Oh, yeah.
Okay. All right.
There we go. Sorry about that.
Yeah, so that is really it's really amazing because I obviously working on POL2, I love the CTD. It's one of the most amazing protein domains like ever. Maybe we can give a little more introduction for the audience who are not accustomed, but this is something that is so unique. I mean, the main machinery of POL2 has this 52 haptide repeats. So these are 7 amino acids that get repeated 52 times. And they are subjected to all kinds of phosphorylation and even more. But phosphorylation really is the key event, and there are different kinases and different phosphatases working in here. And this is critical for the whole transcriptional machinery. And it's so hard to model. It's just like so hard to model because this is such a flexible domain that obviously was never, fully captured in any structure. So, I mean, what Yuan has been able to start giving us the flavor of how things are happening, structurally, it's amazing. So this is what really excites me incredibly about this work. And what I want to ask you on, I mean, again, talking about the CTD, first of all, if you think, so what do you think is the threshold of phosphorylation in order for mediator to start losing affinity, right? Because mediator has this great affinity for the unphosphorylated form of the CTD to kind of get this whole machinery starting. And if you think that, I mean, I would potentially think that when mediator latches off, this is the time when the CTD gets handed over to the integrator complex, right?
That's right.
Probably. Though I still keep wondering, if in principle, because of the multiple repeats, if we could envisage a situation in which both the mediator and integrator are sort of latching onto the same CTD at any given time during the transcription cycle. So what are your thoughts about it?
Yes. So about the timing of kind of event of during that kind of narrow window of event, initiation to elongation transition. So one thing we notice is that for the mediator interface with the CTD, clearly a phosphorylated CTD is not going to be compatible with the mediator. So that's a very strong argument. Once you gain enough threshold of the CTD phosphorylation, that's going to be a very natural signal for mediator to allow it to disengage with the polymerase. And so based on the structural snapshot we captured, what we only be able to say is that we don't know where, because we don't see a continuous density. The only thing we see is two discrete density corresponding to two different segments of the CTD. One is likely to be very, very close to, say, the very first few CTD that has not been phosphorylated. And we think that that region of CTD is probably not going to be ever phosphorylated. That's kind of works as a model to stabilize the interaction between the polymerase and mediator. Whereas the rest of the 52 repeat, or 50 also repeat, is going to be continuously phosphorylated until you gain enough threshold that is going to be able to compete with the footprint of the CTD on the mediator. So so far, we don't know what's actually the trigger of this translocation. There's one possibility in the initial submission phase of the paper we included, but it's too hand-waving, but I think it's maybe more fun and okay to talk about it at this kind of a setup.
Oh, yeah, absolutely.
Which we think one possibility is while POL2, maybe along with mediator, is to so-called clear the promoter. It's basically threading in DNA into the active site to allow scrunching of the DNA. There are evidence, biochemical evidence from Kenji Murakami's lab, indicating that similarly to the yeast system, there's a possible, nearly 100 base pair of DNA being scrunched and producing RNA without disrupting the initiation complex. So that process would likely to allow you to thread the DNA into the active site, but at the same time allowing enough time for the CTD to thread in through this narrow channel to allow this kind of a progressive phosphorylation event to happen. Yeah. And then at the time when there's enough CTD being phosphorylated, you will the DNA at the upstream is going to close up to allow the full bubble collapse. But at the same time, that will be a signal for the CTD to totally disengage with the mediator as well. And we have not talked about potential phosphorylation event on recruitment of capping enzyme, co-transcriptional splicing machinery. I think that there's all those things happening in a series event to allow you to load the proper machine to take care of the processing of the RNA.
Yeah, absolutely. No, it's great. Well, it's funny that the integrator complex has affinity also for unphosphorylated CTD. So I kind of wonder if there is, this, which is why I ask, like, who knows, like, could we possibly think that both mediator and integrator are actually bound to a CTD at the same time, non-phosphorylated CTD at the same time, or could that actually be competition? Even though I don't know what would that kind of competition bring on? It would essentially, well, mostly I guess prevent initiation from happening, right?
Yeah, so from structural point of view, there's this almost consensus in the field after seeing all the elongation complex snapshot has been captured mainly by Patrick Kramer's lab. And there's this very interesting kind of one-to-one interface. If you view POL2 as kind of a central enzyme, you have a series of initiation factors, mainly the neurotranscription factors mediator, being almost coating the entire surface of the polymerase. And then if you compare it to the elongation complex, the same surface is basically being replaced by, say, PAF1 complex, DSIF, NELF, those kind of factors. So it's very unlikely that mediator will still be able to engage with the polymerase while the pausing is already happening. So there's some kind of a handoff, mostly due to the steric clash, essentially.
So this has been really cool, and I know we're running up against time. So I wanted to ask you, if it's okay, kind of a different question. If you think about like somebody who's being treated for cancer, or maybe they've got a family member who's getting treated, and maybe they're thinking like, what caused this cancer? Why did it spread? How could we have prevented this cancer or caught it earlier, or how could, treatments improve. Where does your work fit into this, and why the work that you just published, from the perspective of a cancer patient or survivor, a caregiver, why is this work exciting?
The work is exciting. I'm sorry, I jumped in. I'll go first. The work is exciting because every single exciting discovery in cancer biology started from understanding the basal mechanisms of biology, such as transcription, right? I mean, like even I'm talking back 30 years ago when, people were studying leukemias because in a way it was the most approachable sort of kind of tumors to study, to get samples and to see what really was going on genetically. And then people started looking at these famous translocations back then, and they could understand nothing up until they started getting, okay, these translocations, they involve transcription factors, they involve MYC, for example. And what is MYC doing? Why is this important? Why is this generating this highly aggressive leukemias and lymphomas in these patients? Because then people started to get passionate about transcriptional regulation and understanding, where all these pieces fit in the larger scheme that is, getting this thing accomplished, getting like a whole process of transcription through. So this is why it is exciting, because knowing, I mean, we're getting to a point, especially look at Yuan's work, we get, we'll really get the structural details sorted out in a way that was unimaginable to just about anybody up until 10 years ago. And this is amazing because we know more. We know structural details that we can, start designing drugs if needed, because we have all of this underlying information. So this is what makes this kind of research so special to me. And as I mentioned, this idea that has been put forward about tumors being addicted to transcription. So they're basically, some tumors, they don't need that particular nasty mutation that makes them unique, but they just exploit what is there, the machine that's available to any other cell, but they hijack it and they exploit in a way that's unthinkable of for like physiological conditions, right? Well, I mean, we may at some point also, which we're also doing, I think, these days, we're using drugs, right, and designing drugs that potentially target pathways that are physiological in nature. And yet these drugs, they don't do that much harm to a normal tissue, but they are able to hit a growing tumor real hard. So this is, in a nutshell, why this is so important, I think.
Yeah, that's great. And I'm going to basically add, from a structural biology point of view, we're in terms of cancer biology, we're thinking what would be, how to differentiate cancer cell versus normal cell. And in most contexts, as Alessandro just mentioned, the cancer cell really rely on a high transcription, all these central dogma processes to allow you to basically infinitively grow themselves, copy themselves. And that's why it's so important to understand these processes, these complicated processes, in a greater detail, and to the detail that we can actually see individual atoms. And that will allow us to act, or scientists, or pharmaceutical companies, to really design strategies to target these processes, these weak points. Because without seeing these details, you will only be able to know, what is involved in this process, but you don't know what is the weak point that you can actually interrogate. Exactly. I think the combination of discovery science, what Alessandro is doing, in combination of visualization tools that we are developing, and allowing us to observing these processing at great details, really is a beautiful combination of strategy to allow us to design next generation therapeutics to specifically target, the weak point of cancer cells. And that kind of apply to, our work in gene regulation, but also another topic, DNA repair, double-strand break repair, which, similar thing. People know what the players are, but in order to be able to really understand these processes by visualizing at great detail is, kind of the ultimate way to understand these these large machineries.
Yeah. Wow, pretty fantastic answers from both of you. So thank you. Thanks for your time and for taking us through your work. This was really fun. I appreciate it.
Thank you.
Yeah, thank you again.
All right. Well, you take care.